Welcome to the Optimizing Analysis Settings with Mutation Surveyor webinar. My name is Phuc Nguyen, and I will be the presenter for this webinar. This webinar will focus mainly on the process settings used to run the analysis and how it could be used to analyze certain sequences as well as solve some issues. For all that concerned, please refer to the specific webinar. We will first talk about how to import data into Mutation Surveyor using two methods. Then we will go into the Contigs Settings and how they can help group problematic data into correct contigs. Overlapping amplicons or low quality data are usually the cause of these grouping problems. For the mutation detection threshold, we will talk about how some of the parameters that can affect mutation calls and what will happen if we change it. The sensitivity can be altered to allow Mutation Surveyor to detect low-frequency variant such as somatic mutation. For explanation of each individual threshold and what they mean, please refer to the introductory webinar for Mutation Surveyor. In addition to low-frequency variants, hypervariable sequences are also sometimes hard to analyze due to the huge amount of variation. There are some settings in the software that can deal with some of the issues that hypervariable sequences present. We will also talk about an option known as base patch, which is used to correct for mobility shift and recall the end bases using a unique algorithm to better align the sample sequence and improve mutation calls. Mutation calls in the software has different parameters for one direction and two directional data. And the final topic that we will discuss is the methylation settings and analysis requirements. There are two ways that you can import data into the software. The first is to use the File drop-down menu and selecting the Open File option. The second way is to simply click on the folder icon located under the File tab. Both methods will bring up an Open Files dialog box in which you can use to import data into the software. After loading your data and running the analysis, you can check if the grouping of your samples are correct by expanding the contigs in the browser pane. This is a BRCA1 dataset that contains overlapping amplicons. As you can see in the browser pane, the groupings of the samples are incorrect. By default, Mutation Surveyor will try to group each sample files by aligning them to a reference file. However, when you have overlapping amplicon like this dataset, it becomes difficult for the software to know which contig to place the amplicons in. As you can see, 10A and 10B samples are grouped into the same contig, and each pair of samples are also grouped wrong. We can check how much overlap these samples have by going to the Project Display Report. This report can only be accessed through the Graphic Display Report from the Display tab on the main menu or selecting the icon shown here. The Graphic Display Report is an alternative graphical view to the main analysis window. In this view, you can see the amplicons and where it aligns relative to the GenBank file for each contig. You can access the Project Display Report from the icon shown here. The Project Display Report is also a graphical display, but it shows the entire project aligning to the GenBank instead of individual contigs. In this report, you can see that some of the amplicons overlap one another. The yellow bars are the amplicons, and the pink bar is the ROI. For more information concerning the Graphic and Project Display Report, please refer to the Reviewing and Editing Data webinar. We will now move on to some of the options to group the forward and reverse pairs of samples into correct contigs. There are options in the Contig tab of the process settings to help the software group the samples correctly. If your sample overlaps significantly, deselect force into one contig. In addition, the exclude first base difference should also be selected, and the threshold should be estimated to the number of bases the amplicons overlap. I set this at 100 as this will prevent Mutation Surveyor from trying to force same direction samples into a single contig when the first base or each trace aligns with more than 100 base difference relative to the GenBank sequence. If you have stringent nomenclature for your samples, then you can specify the characters that you want to group by. I am specifying the characters from 14 to 16, which in this case are the exon number for individual patients. You can also use the exact file name match options to group samples by their names. Both options in the exact file name match requires that the nomenclature of your samples be the same if you want to group them together. With those options selected, I ran the analysis and the groupings of my forward and reverse pairs are now grouped correctly and placed into their respective contigs. In this zoom view, the 10A groups are in one contig and the 10B group are in another contig. If you still encounter problems adjusting the settings to group your amplicons correctly, make sure the character you specify are in the same position in all samples. We will now move on to the mutation detection threshold and options. Under the Mutation tab of the Process Settings, you can see all the thresholds set by the software for mutation detection. These default settings have been optimized to detect variations in dataset fairly well. However, all of the threshold can be changed if needed. For example, if you want to make your analysis more stringent, increase the parameters. If you want a threshold to be less stringent to allow for high sensitivity for germline mutations, then decrease the threshold. By lowering this threshold, you are lowering the stringency and increasing the sensitivity to detect mutations that appear below normal threshold, such as somatic mutation. At the same time, by lowering all the threshold, you may also be picking up false positives. In addition to those settings, you can also use a check 2D small peak option to tell the software to look for wavelength with intensity above 200 RFU of the same color and the same position in both the forward and reverse strand. This is usually a good indication of low-frequency variants. For more information concerning the detection of low-frequency variant, please refer to the webinar. In the same tab, the mutation call threshold defines a parameter for a failed mutation call, low-confidence mutation call, and high-confidence mutation calls. The passing parameters in this tab is the same as the one in the Mutation tab as described previously. Any mutation with parameters below the fail threshold will not be called. Any mutation that falls between the fail and pass threshold will be color-coded red for low confidence. And any mutation that passes all the threshold will be color-coded blue for high confidence. This color coding scheme is shown on the mutation peak as well as the mutation table. This is a set of data for the TP53 gene. The 2 mutation call has blue text, which shows that they are high confidence and all parameters are well above the set threshold for mutation calls. This is also denoted in the mutation table at the bottom. Within this analysis, I have also lowered the threshold and used to check 2D small peaks. Along with the 2 mutation detected in this sample, there are also green bars along the electropherograms that indicate the possibility of a low-frequency variant such as mosaicism, heteroplasmy, or somatic mutation. This type of analysis is further elaborated in the low-frequency variant detection webinar. We will now move on to options that involve hypervariable sequences and low-quality trace. In addition to the threshold, Mutation Surveyor does many calculations in the background when processing one-directional and two-directional data. In the mutation detection area, the high sensitivity and medium sensitivity pertain to these internal calculations. High sensitivity will pick up almost every variations in the data. This setting will apply less stringent calculations to the variation. This setting is good when you want to call everything, even false positives, so that you can review the data more thoroughly. If you are concerned about the high number of false positives in your project, you can select medium sensitivity in order to make the parameters more stringent. The Allow for Hypervariable Mutation Detection settings, when activated, will extend the start and end region of the comparison region if 2 conditions are met. The first is that 4 out of the 10 bases outside the comparison region have a greater than or equal to Phred score of 15. The second condition is that 8 out of 10 bases outside the comparison region must match to the reference. By extending the region out further, more potential mutation can be called. This option is especially useful when analyzing viruses or samples that contain high mutation frequency. The Delete Indel option will tell the software to automatically delete any indel calls from the project. This option helps to eliminate false positives that are common to hypervariable sequences. The Allow software to delete mutations will let Mutation Surveyor delete illogical mutation that has a high background noise of greater than 250 RFU. This setting can be used to eliminate some false positive if your data is affected by noise. The DieBlob baseline removal option will tell Mutation Surveyor to conduct another set of algorithm to detect broad DieBlob peaks and eliminate the baseline problem caused by the DieBlob. To better illustrate how some of these options works, I will give some example. In this example, the illustration will show the difference between DieBlob removal activated and inactivated. Although dye blob artifacts are becoming less of a problem due to advances in dye chemistry, they can still cause major problem in Sanger sequencing samples if it occurs. Using the dye blob baseline removal option in the process settings, it will automatically remove the baseline problems caused by the initial dye blob as well as remove the dye blob itself. This will eliminate the false positive mutation calls due to the original elevated baseline problem. As you can observe in the right-hand illustration, most of the false positive mutation calls are in gray. This means that the setting Allow Computer to Delete Mutation is on, and Mutation Surveyor recognizes these peaks as having very high background noise and considers them illogical.